Hello, my name is Sara, a student of Queensborough Community College. I am presenting a mini review on how gene functions are studied in different organisms. My mentor is Dr. Andrew Wen. Here is the poster of the mini review discussing various methods of studying gene function in different organisms. Gene expression is a process by which information encoded in the gene is translated into protein. The type of protein determines the fate and the function of the cell. Gene expression is a highly regulated process. To understand the function of the gene, scientists have exploited the gene mutations and evaluated the phenotypes in the absence of the gene. I will briefly discuss how genes are studied in different organisms. Gene function in prokaryotes has been studied using E. coli. Of course, many different prokaryotes have been used, but the original studies using E. coli has shifted the paradigm in the way we study microbes. The right-hand image shows a method called replica plating done by Joshua Lederberg and Esther Lederberg, which helped identify auxotroph mutants. Tryptophan pathway is an interesting cellular metabolic pathway by which gene function of E. coli can be studied. Auxotroph mutants are those that have a mutation in gene encoding an enzyme necessary in the biosynthesis pathway of tryptophan. This method involves growing a population of E. coli bacteria that were mutagenized on two separate plates, one with complete nutrients and one lacking tryptophan. After comparing the growth on both plates, the auxotroph mutants are identified as the growth present on the plate containing tryptophan. Once the auxotroph mutants are identified, a complementation study can be done. This means that they can transform the auxotrophy with the E. coli plasmid library and identify the gene that can revert the auxotroph mutant back to the wild type. This can also be done with other prokaryotes. C. elegans is a nematode used as a model in 1960 by Brenner. Because many genes in this organism have functional counterparts in humans, they are studied to understand human diseases, particularly neurological defects. There are mutant stocks available for one to study or knock down the gene using RNA. To study gain of function, the heat shock promoters are fused upstream to the gene of interest. After acute heat shock, gene expression is induced. This organism has been used to study genes associated with asymmetric cell division of apoptotic genes. The organism readily takes in RNA, genes associated with aging, and polycystic kidney disease. Xenophus is also known as a frog. Gene function is analyzed by studying their embryos. Underexpression or overexpression is done by gene gain and gene loss strategy, as shown on the image on the right. The embryos of Xenopus is easily injectable with cloned cDNA, mRNA, or antibodies. DNA injection is done to study overexpressed extracellular protein and antibodies to disrupt the normal protein function. Gene function in Drosophila is also known as a fruit fly. It's studied through mutagenesis, transposons, and transgenes. In mutagenesis, mutant lesions are created with the help of chemical mutagens like ethyl methane sulfonate. Some transgenic methods that are used consist of transposition and site-specific integration at ATTP sites on the entire genome. Drosophila is an ideal model system to study because many genetic mutations can be observed with the naked eye, since the female can lay 30 to 50 eggs a day. Thus, it can be used to study genetics and development. Mus musculus is also known as the house mouse. One method of studying gene function in Mus musculus is gene knockout, either using embryonic stem cell growth on a feeder cell and allelic exchange of targeted DNA region, or more recently using the CRISPR method. Another method of studying gene function is to overexpression the gene of interest by injecting DNA into a fertilized oocyte and later transferring that oocyte into a pseudo-pregnant female. The ultimate goal of scientists is to obtain the gene function of the entire human genome. The study of gene function is highly essential for genealogy, which is the study of the descent of an organism from its ancestors, forming a family pedigree. The significance of genealogy is that it helps to provide information about genetic diseases such as sickle cell anemia. Through the help of studying gene function in humans, scientists are on the path to use CRISPR to correct the genetic defect. CRISPR is being used to treat cancer, type 1 diabetes, blindness, cystic fibrosis, Huntington's disease, and Parkinson's disease. In all these cases, the patient's cells are removed and modified outside the body before giving back the patient themselves. Recently, it was reported in the news that CRISPR can be injected directly into the blood to treat genetic diseases that cause misfolded proteins. This idea was modeled for mRNA injection for COVID-19. Here are the references used in this mini review. I am highly grateful to the CUNY Research Scholar Program and my professor, Dr. Andrew Wen, for providing me this great opportunity. Thank you for all your time, and all questions are welcome.